The chemical equation for photosynthesis with the help of light energy is CO2, carbon dioxide, plus H2O, water, yields C6H12O6, glucose, plus O2, oxygen gas.
The picture you are looking at is a structure of a chloroplast. A chloroplast is a green organelle that catches light through the site of photosynthesis, which creates organic molecules as food for the plant. There is an outer and inner membrane. It contains the granum, which is a stack of thylakoids, and a thylakoid is a flattened sac, which the light reaction goes through. Also, the jelly-like substance in between the granum is where the Calvin cycle takes place. The inputs of the light reactions is the photon of light and water. The outputs are O2, ATP, and NADPH. The inputs of the Calvin cycle is CO2. The outputs are G3P, which is glucose, ADP plus P, and NADP plus P.
The picture you are looking at is the structure of a leaf. It has many parts that help the leaf stay alive. The cuticle is a waxy-like substance that prevents the leaf from desiccation or drying out. The upper epidermis and lower epidermis protect the cell from injury. Palisade mesophyll is the greatest site of photosynthesis. Spongy mesophyll allows carbon dioxide to enter through the stomata and water to accumulate through the air spaces. The vein is made of xylem on top, which carries water from the roots to the leaves so it may be stored. And phloem on the bottom transports the organic molecules that are made in photosynthesis down to the roots to be stored until they are needed and used in the winter. Guard cells open and close the stomata due to hypertonic and hypotonic solutions, allowing oxygen to exit the leaf and carbon dioxide to enter. Lastly, the stomata is the pore inside the leaf that allows gas exchanges.
This is light reactions. Water comes into the chloroplast and splits into three things: oxygen, hydrogen, and electrons. The oxygen combines with another oxygen and leaves through the stomata. Hydrogens accumulate in the lumen. Electrons are transferred to photosystem 2. Light energy excites the electrons and moves to the long ETC, while the electrons are moving from photosystem 2 into photosystem 1. Next, they actively transport more hydrogen into the lumen, becoming weak. Electrons then move into PS1. It gets re-energized with photons of light energy. The electrons then move to the short ETC, where they enter the NADP plus reductate. The previous energy for NADP plus and hydrogen combine to make NADPH, with the electrons traveling to the Calvin cycle. The hydrogen that accumulated in the lumen funnels through the ATP synthase, giving energy to combine ADP and phosphate to create ATP.
Chlorophyll A is a pigment and is the main part of photosynthesis. Chlorophyll B and carotenoids are pigments that transmit energy to chlorophyll A. Chemiosmosis is when hydrogen funnels through the ATP synthase. It gives energy so that ADP can combine with the phosphate to make ATP. It's over. Chemiosmosis is when hydrogen funnels through the ATP synthase. It gives energy so that ADP can combine with the phosphate to make ATP.
This is the Calvin cycle. In the Calvin cycle, you start with 3 CO2 entering one at a time that combine with RuBP, which is a 5-carbon compound at the riboscope. It then becomes a short-lived 6-carbon compound that immediately splits into 6 GPAs. ATP that was created in the light reaction then comes in and adds a phosphate to each PGA, making the PGA a biphosphated PGA. Then NADPH that was also created in the light reaction comes in and takes away the phosphate that ATP just added. The ATP turns back into ADP and NADPH turn back into the NADP+, both of which go back to the light reactions to reproduce. 1 out of the 6 G3Ps exit the cycle and create half a glucose. The other 5, with the help of 3 ATP, become RuBP and the cycle starts over again. The cycle continues until 1 full glucose is created. When the cycle has created enough G3P to create 1 full glucose, The glucose can then become a protein, fat, or carb, which are all macromolecules of life.
The environment can change the rate of photosynthesis in many ways. One way is light intensity. The greater the light intensity, the greater the rate of photosynthesis, but it plateaus at a certain amount of light. Another is temperature. It helps photosynthesis to a certain point until the integral protein denatures.